The nucleus of a human cell contains the pairs of chromosomes shown here. The Rett disorder results from mutation at any one of over 100 places in the gene MECP2. That is the short name for the gene which makes the protein methyl-CpG binding protein 2, located at the tip of the long arm of the X chromosome, XQ28. In every cell, a female has 2 X chromosomes, one from each parent. A male receives the X chromosome from his mother and the Y chromosome from his father. Most mutations occur in the reproductive cell of an otherwise normal parent, more often in sperm than in ova. Therefore, the child receiving the X with the mutation usually receives it from the father and is female. Less often, the mutation occurs in an ovum, in which case the child who receives it may be male or female. Although all the cells in the female body contain 2 X chromosomes, one from each parent, each cell uses only 1 X, the other being inactivated. Most females use the X from each parent equally, so in Rett disorder, half the cells are using a normal X and the other half a faulty X. This situation typically leads to classical Rett syndrome. Rarely, a female uses far more of one X than of the other. This is called skewed X inactivation. If most cells use the X with the mutation, then the condition is likely to be more severe. If few, then milder. Since a male has just 1 X chromosome, if that has the mutation, every cell is affected, and the condition is severe. Rarely, a mutation arises after the embryo has begun to develop, when it affects only a portion of cells, and the severity is less. This young woman has very mild Rett, as almost all her cells are using the normal X. However, because she does carry the gene with the mutation at one of her X chromosomes, she may still transmit the mutation to her offspring with a 50% risk to any child. Here her son and daughter both acquired the disorder, the boy with severe Rett and the girl with classic Rett. The mother and both children have the same mutation. In Rett disorder, mutations have been found at all the places indicated here by arrows. Some locations and some types of mutation are associated with milder or more severe disease. For example, R133C and R306C are usually associated with milder disease. T158M, R255X, R168X, R270X with more severe disease. The MECP2 gene produces the MECP2 protein through a transcription process. The gene becomes active, we say expressed, where it is required, in response to local conditions in the body. The 2 marked areas are the methyl-CpG binding domain, MBD, and the transcription repression domain, TRD. These are known to be important for the function of MECP2. In healthy cells, these domains regulate the activities of other genes by preventing their transcription into active proteins, effectively silencing their activities when they are not required, and thus orchestrating the activities of cells. A mutation interferes with this process. Here is a graphic image of the MBD domain showing common mutation sites. Here are some important genes whose activity is known to be affected by failure of MECP2 in Rett. Professor Angus Clark, based in the University of Wales, has provided genetic services and conducted research on Rett. We asked him when genetic testing would be advised.
I think it's very fair for the clinician to go ahead with that once it's occurred to them, really, as long as they're taking the family with them. And occasionally there'll be a family who to whom it has not yet occurred that the child may have a really serious disorder, and then there might be reason for suggesting the diagnosis and talking about testing before doing it. But really, as long as the family are clear that they want a diagnosis, then as soon as it has occurred as a possibility to the clinician, there's no reason not to do it.
We asked if he could give a general figure for risk of recurrences.
There is a risk, because we know there are very occasional families where it has happened to more than one child, but we know that the risks are small, no more than a few percent. In the situation where a woman carries the mutation, then the chance of another child inheriting it would be effectively 50%. So that would be 50% of daughters inheriting Rett syndrome. And for males, the risk would probably be of something more severe, giving rise to a child with very severe illness from soon after birth, but having the same cause.
Mutations are identified by a combination of tests, and new methods are still being developed. So far, mutations have been identified in almost 90% of people with classic Rett and 40% of less classic cases. Previously unrecorded mutations are still being identified. Although in most families recurrence is unlikely, mutation testing is advisable to provide reassurance and to detect mildly affected carriers. Prenatal testing is possible if the mutation is already known.